Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment and thank our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Mark your calendars for July 21st, as we will be hosting another free symposium in the ecosystem, this time on the molecular and structural insights of GPCR activation. Our program includes talks from Katerina Nemec, Alexei Serbo, and Polina Isankina. We're taking you on a structural and molecular journey to understand GPCR activation better. If you'd like to give a talk, we are still constructing the program, so please reach out by email at hello@drgpcr.com. We also set aside 2 hours for poster presentations and networking on a new space called Kumo Space for this symposium. There is no limit on the number of posters we can accommodate, and everyone is welcome to present a poster at our upcoming events, including the one on July 21st. We will not be selecting posters. You will not have to send in any posters. We only ask you to fill out the poster form so that we can capture your information. Part of that information is a 1-minute video abstract that we will share on YouTube. We'll also share it on our website so that people can see what you will be presenting about. You can also join us live for any of our events by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. For more information and an updated schedule, you can go into the ecosystem. If you haven't noticed yet, we've added menu buttons at the top of our page for your convenience to help you navigate the ecosystem more effectively. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today, and I was going to say this morning or this evening, depending on where you are, I'm excited to have with me Brendan Wilkins. Brendan is finishing up his PhD in Nicholas Smith's lab in Australia, so it's almost your bedtime, and my morning just started. Welcome, Brendan.
Thank you, Yamina. It's such a pleasure to be here.
I'm excited to have
Thank you
you
for
on
having me.
today. Thank you. And there is a delay due to the big distance between us, so please excuse a little overlaps. But how about we start you off with a quick introduction as to who you are and what do you do currently?
Yep, so as you just said, I am a final year PhD student in the Department of Pharmacology in the School of Biomedical Sciences at UNSW Sydney in Australia. So I would class myself as a molecular pharmacologist. I'm very into the technical assay design, plate-based assays, and everything like that. So everything molecular pharmacology, of course, surrounding GPCRs is very interesting to me.
That's my second sip of coffee today. You're mentioning GPCRs and obviously plate-based assays. I really loved your talk at the last symposium, and I thought it was
Thank you.
so well presented, so methodical. And I do have I also think of myself as a molecular pharmacologist, and everything that's plate-based, cell-based, live cell assays is just a phenomenal. I think it's so nice to be able to look at that type of data. So how did you get to where you are today?
Um, it's been um, an interesting road. I remember as a kid, I this is a distinct memory of mine. I would get a bucket from the laundry and I would find all of the little things in the bathroom cupboards and things that I could mix together, which in retrospect may have been a little bit dangerous. But I remember rifling through my sister's talcum powders and everything like that to try and make the most interesting concoction I could. So I think there was a scientist in me from a very young age. But despite those behaviors, I actually wanted to be an architect. Um, and I think this was mainly from watching fantasy movies and TV shows and things like that that had these amazing buildings, and I wanted to design those things and and fall in love with buildings like that, but I didn't actually know how an architect would do that. And it wasn't until a work experience placement in high school that I spent a week with an architect and found that I really did not want to be an architect. So so yeah, later in high school, I was taking science electives and found that I was really interested in biology and chemistry. And um, those subjects were what piqued my interest most in high school. And I actually applied into medicine um, to university after high school, but I didn't get in. And thankfully, because also in retrospect, I definitely would not have liked to be a medical doctor. So I ended up in a Bachelor of Science, and I grew up in a rural town, and so I didn't really have much background of what university would be like. There wasn't much industry, science industry in my hometown. There was a small university, but the focus wasn't science. Their focus was communications and media and journalism, drama, um, things like that. So I, I came into university with, um, I guess, a naive mindset. I didn't really know what I was going into, and I was only doing it because I was interested in doing it, and I didn't have anywhere else to be taking my life. And so I started majoring in chemistry actually because that's what I did best in high school. And I quickly found out in second year that chemistry was not for me, even though I was enjoying the content. And I, from the undergraduate subjects that I've done, the only majors that I could then go into were pharmacology or physiology. And so I jumped ship into those, and I think that was a very serendipitous thing to happen because I just fell in love with pharmacology and physiology and everything everything disease related from a scientific perspective rather than a human population or public health perspective. And I'm so thankful that I realized that I was having a really difficult time in chemistry and found my way into pharmacology because I haven't looked back. It's been one of the most incredible, um, I guess, career choices of my life. And now I find myself doing a PhD and hopefully will contribute some great things to the field.
Thank you for that. It's funny because you were mentioning being a child and trying to make concoctions, and I was gonna say, oh, that sounds to me like a chemist. But then
Definitely. Yeah. Yeah. So I dabbled. I tried to be a chemist, but yeah.
I
Instead now I put all the concoctions onto live cells and see what happens.
and it's interesting that you also meant I absolutely can relate to what you were mentioning, not necessarily knowing which way to go, and also the fact that you realize that chemistry is not for you. It's not something that you really enjoy, and medical school would have not been something that you didn't that would you would enjoy. And I think it takes a lot of courage to say, well, actually, this is definitely not what I want to do, and I'm going to do something that I do like instead of pushing forward in a field where you're not happy and you're not
Yeah,
enjoying.
it's a massive life lesson. And just retrospectively, I guess I used to think that if you made a mistake, then it would be the end of the world, and it's just
Yeah,
an anxiety thing. But really, it's a learning lesson to go through those things, to have a difficult time, and to come out of it stronger and knowing what you don't want to do.
yeah,
It's much like research in itself having a lot of negative results. The negative results are still just as important as the positive results.
yeah.
They still tell a story. Um, they're very important.
I think so too. I think so too. And I think it you touched on an interesting point around accepting failure. And I think when you're in a difficult position, no matter what that position is, the first instinct is to say, this is the end of the world. Why am I in this position? And I think at that moment, although it's difficult, but telling yourself, actually, no, this is a learning opportunity. Now is the time for me to figure out how do I react. And next time, if a situation like this arises again, I know what to do or what not to do in this
Exactly.
case. So we
Yeah.
definitely skipped to the advice for to young
Oh,
scientists.
absolutely.
Now I'm
But
no,
I
I
guess
think
there's
it's
plenty
great.
more of that.
Exactly. I think it's great. I think it's important. Uh, on my end, I totally, again, I can relate to this. And it's interesting because this is our first time having a conversation. We met briefly at the symposium, but when I was in college, I did my French, I failed the French class. And I thought it was the end of the world. And it was just a matter of 5 points or 2 points or something really stupid, basically. But then I ended up retaking the class with a new professor, and it was such an amazing experience. It didn't change, nothing changed. The world did not stop, and my career didn't suffer from it. I still graduated on time. It's a little bit more effort to finish up, but hey, it's not a problem anymore.
It's funny you say that actually, because while I was majoring in chemistry, I was taking a minor in French as well. So I was doing university level French, and I started off from introductory French without knowing any French at all. And it's something that I really struggled with. Um, and I went very close to failing, I think one of my intermediate French courses. And and I think that was part of why I was not enjoying my other courses so much, because I was finding university difficult in general. Um, but yeah, it's funny that that similarity is there.
We have that in common as well. You mentioned moving from a small rural town to the city to the university. How was that transition for you?
Yeah, it was I was I had a lot of support. Um, I knew that I wanted to move out of the rural town, and that I wanted more than what that town could offer me. And so when I moved to Sydney, I was 18. I was straight out of high school. Um, None of my family lived in Sydney. Um, and so I'd moved into a dorm. I
Mm.
think you
Yeah,
guys call it, but we
yeah.
we call it college.
Hmm.
So we call university as a whole university,
Hmm.
and then the colleges on the university campus are the dormitories.
Okay.
So I moved into a college, and I made a lot of friends, and I'm still friends with a lot of those people today. And I'm really thankful for that, because they are my tight friendship circle in Sydney. That will, I imagine, be my friends um, for a long time um, as well. But I think my main difficulty was not being prepared for uni and what university would offer, because it's an entirely different learning style to from between high school and
Hmm.
university. And I hadn't been prepared for that. And so I went to maybe 2.5 years struggling um, without that information and without that skill set to learn independently and such. But it was something that I learnt along the way. And I took one semester off after I decided that a major in chemistry wasn't for me. And when I came back to a major in pharmacology and physiology, I had developed that skill set to study properly, to take responsibility for study, and to make sure that I got good marks. And I think that in taking a major in pharmacology and physiology, I had much better mentors than I had in chemistry. So that was a massive help. Just mentors to inspire you to learn, to inspire you to be passionate about something. And I guess we'll get onto that in a minute, but yeah, my mentors in pharmacology and physiology were really important. Um, yeah,
You made an interesting point mentioning how difficult it was for you to go from high school to university because there were skills that you were not taught. And I think that's a big issue in general with the educational system, is that there are skills that we are not taught in school. We're taught to obviously read, write, and count, and many more, but we are not taught the basic skills as to how do you get organized? How do you learn to study? Figure out what is your style of studying. What are the things that you should be focusing on when it comes to communication? What is your communication style? And help the younger generation develop, get self-knowledge. Because if you know that you're a visual person, listening to something is not going to work out. So you directly go to that visual aid. If you're someone,
absolutely.
and I think this is such an issue that I have trouble with in general, because I struggled as well. So in Canada, in Quebec at least, you go from high school, then you go to something called college or cégep, which is 2 years at a different school before you can go to university. So you basically go to university at 20. And that college I went to, it was focused on hardcore sciences, but the difference in level between high school and the college was enormous. And I failed my first exams: math, physics, chemistry. And then I got called into the office to do a test to see if I'm in the right field. And it turned out that yes. And it took me a year and a half after that first semester to catch up. But it was extremely difficult because I was not prepared for it. And there was no support for it either, which I think is also a problem at universities, is that, okay, we're not teaching you these skills, but then we don't have the support. It's kind of a free for all,
Yeah,
which
and
is
and I think
unfortunate.
the issue transcends into adulthood as well. Like, particularly us as researchers, I see a lot of
Yeah.
academics that um, did a science degree. They did their PhD in some kind of scientific field, and then they end up in an academic position where they're expected to lecture, where they're expected to design teaching content, they're expected to run tutorials and design labs for students to do, write exam questions, all of that which they weren't taught how to do.
Yeah.
Their training is as a scientist.
Yeah.
So I think that that is an issue in science, at least, that not all scientists are good teachers, but to survive in an academic environment, you need to learn how to be a teacher in some way. Um, and yeah, it's
No, I agree.
difficult.
I agree. I agree. I think science is hard enough. And as a trainee, as a postdoc, as a young professor, you're focused on getting as much data as possible so that you can get funding, so that you can write papers, get funding, and so on. And you don't have the time nor the support from the university to teach you how to do these things. And I'll
Mm,
take it one step further: mentoring your students in the lab, or even just how to pick students in the lab.
mm,
Based on what? What typically ends up happening is that they pick especially when you're young and you need to fill your lab, you pick anyone who knocks on your door, and then they might not be a good fit, but then I think there is a skill to be developed around management, which also include mentorship, management, leadership, which also includes having difficult conversations. How do you
yep.
tell that student, actually, I don't think you're very good at this, and maybe you should reconsider your career choices in a way that doesn't kill the students' dreams, but gets them to think and then make a plan for what could be better for them. And that's
Yeah, and
such a difficulty.
I think that also occurs at the the point in their life where they're not really sure what they want to do. Similar to
Exactly.
where I was when I was switching my major to pharmacology and physiology. They might not necessarily know where they're at in life. So they need that mentorship to guide them towards
Yeah,
a career path that's going to be fulfilling. Yeah, it's
I
interesting.
think and I think we could be talking about this for a long time. So why don't we get back to GPCRs?
Yeah.
And let me ask you, how did you get into studying GPCRs?
So I this and purely this to Angela Finch. A lot of people in the field know Angela Finch as a diehard GPCR fangirl. But she um, she convened a third year pharmacology course here at UNSW called Molecular Pharmacology. And the course is designed around the molecular pharmacology of GPCRs. So it's everything GPCRs, signaling, um, targeting GPCRs therapeutically, and and it was that course alone that set me on my trajectory of GPCR research. And she was such a great mentor throughout that course that I decided to do honors with her. So um, Nicola's spoken to you about this before, but honors is in Australia is kind of it's a research year somewhere between undergraduate research and a master's degree. So in a lot of ways it's our version of a master's, even though we still do have master's degrees uh, separately. And it was in that honors year that I worked on the beta 2 adrenoceptor and allosteric modulation of that receptor. And I just had such a phenomenal year researching GPCRs with Angela. I remember so many times I would be lying on her office floor having a crisis about why my radioligand binding assays weren't working.
I'm
And
laughing, but I'm sorry. It's I can imagine you being on the floor and being like, why isn't this working?
just
But it only shows that you're very passionate about the science.
yeah, absolutely chaotic scenes here at UNSW, but Angela was always there to help me and to guide me and to troubleshoot with me and I learned a lot about troubleshooting from her just from those conversations where I would yeah be spending time in her office with her. Um, and she supported us, the students in the lab, to go to ASCEPT. So
Yes.
ASCEPT is the Australasian Society of Clinical and Experimental Pharmacologists and Toxicologists, and we have a national conference annual scientific meeting every year. And it it's internationally known. A lot of famous researchers are invited to come and talk, and I think it's such an exceptional time event to go to throughout the year. And it was there that I just absorbed all of this knowledge of GPCRs. The PhD student in our lab whose project I was following on from in honors um, couldn't present her work, so I ended up giving an oral and a poster presentation for her at that conference with some of my data included. And I remember it being the most terrifying thing in the world because Brian Kabilka and Robert Lefkowitz were there. It was so intimidating. And during my oral presentation, I was standing up on the stage and I could see Kabilka and I could see Lefkowitz and oh man, the amount of adrenaline that was running through my system. No doubt acting on the beta 2 adrenoceptor um, was just crazy. And they both asked me questions at the end of my talk, and I may have been a little bit sassy to Brian Kabilka, which I cringe. I still cringe about every day. But it was such an exciting experience, and um, yeah, GPCR research is I think any research can foster that environment, but I think I'm particularly excited about GPCRs for that reason. And after that, I didn't really know what I was going to do with my life yet again. I guess that's a common theme in my life. But yeah, so
You're,
I
you're not alone.
yeah. Um, so I chose to do a research assistant role with a, a a PI here at UNSW. He did have one GPCR project. His name was Nigel Turner. His name is Nigel Turner, and he runs a mitochondrial bioenergetics lab where he focuses on particular enzymes involved in um, mitochondrial bioenergetics. And
Interesting.
I worked with him for a year, and at the end of that year, I went to one of Angela's um, honors students' final seminars,
Mhm.
and of course, it was surrounding GPCRs. And Nicola Smith was there examining the students. And after the session, she walked up to me and she said, "Brendan, what are you doing in Nigel's lab? He doesn't focus on GPCRs. How about you come and work for me?" And so, of course, I said yes, and I got sucked back into the GPCR world. And so I went and worked with Nicola at the Victor Chang Cardiac Research Institute for 2 years, where I worked on GPR 37L1, the fabled GPR 37L1. Um, and yeah, I guess my love for GPCRs was reignited, and I moved over to UNSW with Nicola to start my PhD in 2020, which was unfortunately exactly when COVID happened, which was incredibly poor timing. But I find myself here now working on orphan GPCRs and still absolutely in love with them.
That's phenomenal. And you mentioned not knowing exactly where you wanted to go. I don't know if you had a chance to listen to the podcast episode where Brian Airy is interviewing me, but I ended up doing a bachelor's in biochemistry because it started with B and not C for chemistry. And I'm so happy that I went with that because I think I would have been miserable in chemistry. But, it took a long time to figure out what I wanted to do. For a long time, it was, oh, I want to be a professor. But then I realized, no, actually, really not. And it took COVID and I want to say less than optimal work experience to start Dr. GPCR. So embrace those moments when things are not going well because those are the, I think, life-defining moments.
Absolutely. Couldn't agree more.
Were you a part of Nicola's lab when this uh, whole story broke with her figuring out that there was an error? Um,
Yeah, I was. So I think that might have been my first year with Nicola when we found when the honors student and her postdoc found the issue, and I was part of the team that had to recover everything. Um, we got the new construct in, and we were madly trying to retest everything that we'd already tested
yeah.
at at the other construct. Um, but we were testing it at the correct newly purchased construct. And we spent a lot of late nights retesting all of the ligands and seeing what we could make of the correct plasmid. But yeah, it was an interesting time having just come back into GPCRs and um, having to not deal with that, but kind of experience the turmoil that Nicola was facing and seeing how much it affected her. Um, but really, I think I learnt a lot during that period because um, I could see um, how much it destroyed Nicola,
Yeah.
um, but also the the nobility and the strength and the just the poise that she demonstrated throughout that entire period was phenomenal. Um, and and for her to retract two of her top papers, um, that I don't think a lot of scientists would under the same circumstances, was really inspirational. And I think it says a lot about the state of research,
Yeah.
in general. Um, but yeah, I think that was definitely a career defining moment for me. Rather than trying to be the biggest, most famous researcher,
Yeah.
do anything to get there. We really need to look into the integrity of science and make sure that we're taking
Yeah.
steps to um to make sure that we're doing the right thing.
No, I agree. I agree. As you know, I had her on the podcast and I chatted with her about it. And wow, it was a chilling story. And kudos to her because
Absolutely.
it was difficult, but I think she made the right choice. I think she set an example. And yeah, I think in science in general, I wonder how many people who would have been in her situation would have handled it so graciously and so openly.
Absolutely.
I think
Couldn't agree more.
she's just amazing, and it's such a difficult thing to do,
Yeah.
especially I think when it's your paper and you're the PI and it's an honest mistake.
Mm.
And I think what I really liked from my conversations with Nicola is everything that she put in place in the lab from that moment on to avoid this thing happening again. And it goes back to the universities not preparing PhD students, postdocs who want to get an academic position to what's coming, and universities not having a standard to say, you got here, great, these are our standards, this is how you collect data, this is how you do it because we want to help you, and you as a scientist and you as a PI and the scientific community in general to avoid mistakes like this.
Absolutely. And I think ultimately graduate students are at the mercy of their PI or
Although,
whoever's teaching them. Often it's a postdoc who's teaching them how to
yeah,
collect that data, how to record that data. Um, and not everyone has the same experience. And
yes,
it's just an unfortunate fact that some students are neglected in the lab and don't learn the right way to do things, and often things are overlooked, things aren't recorded properly, and we kind of you can end up down these avenues of
yeah,
mistakes and
yeah.
poor data. I'm very lucky to have been mentored by people like Angela, Nigel, and Nicola throughout my graduate career, and I'm very thankful for that.
What did you what how did this time this whole situation with this error and the retraction influence how you do your work in the lab?
I've always had um, a very keen eye for for everything that I do in the lab. I'm quite meticulous as a person, so just by nature, I
Yeah,
think that I'm very on top of things like that. But definitely, I'm much more controlled when I make a dilution from a maxi prep or something like that. All of my dilutions, I'll know exactly where they come from. I'll have on the tube this was diluted from the maxi prep that was made on this date, for
yeah,
example. So it's kind of like nth degree stuff,
yeah,
um, that a lot of people would view as unnecessary, but it's just a safeguard. And I
yeah,
have so many safeguards in the lab like that, to make sure that this tube came from there, or this drug went into this plate, or
yeah.
things like that. But I think the area that is most affected is um, my data analysis, my file control, my
Mhm.
versioning, and things like that. I think that's something that I didn't really have in my skill set until possibly um, when I joined Nicola's lab and went through what happened. Um, but it's definitely something that I've learned to have a keener um, or pay more attention to.
Yeah.
So yeah, yeah, definitely some growth there.
I think record keeping is so important, yet it's so difficult to do it. One, because you don't have the tools necessarily that work out. You know, you're given a lab notebook. I mean, paper is great, but paper disappears. Um,
Mm,
and I think nowadays, with everything being electronic, it's harder to delete. But then I think it's also important to write down everything that you've done, which is, and I'm saying this knowing that I don't think I was such a good record keeper, because I was always tempted to do the assay and then do some back of the envelope calculation and then stick the back of the envelope in the lab notebook, which is not the right way to do it, because you want to be able to go back 5, 10 years from now, 10 students later, and figure out where did that data come from. You also want to know what was the lot number of the
yeah,
bottle, of the compound bottle, just because you never know. And that's
yeah,
something
absolutely.
we're not taught from the get-go. We learn as we
Yeah,
go.
we work with electronic notebooks now, and I do find it difficult in the lab when you just want to scribe something down back of the envelope. Um, but we do have dedicated laptops in the lab for electronic notebook taking, but it's just not the same as writing things down on a piece of
Yeah.
paper, quickly scrolling out a calculation or something like that. So I've kind of developed a hybrid system where I have a bound lab book and
Mhm.
all of my back of the envelope calculations go into that.
Mhm.
So if I'm doing drug dilutions or something like that in the lab, I will have it recorded properly.
Yeah.
And if it's important, I will take a photo of it and upload it into my electronic notebook.
Yeah.
And and then if it's not even in my electronic notebook, that bound lab book will stay in the lab.
Yeah.
And so literally everything that's recorded is either on the electronic notebook,
Yeah.
it should primarily be on there, but as a fail-safe, it will also be in that bound book.
Yeah.
So I think there's definitely merit to adopting systems like that to avoid overlooking things in the lab.
Yeah. Yeah. Um, I know we've talked about many things so far. Let's get back again to GPCRs. You've worked on the infamous GPCR 37L
One, yep.
one. You've worked on beta adrenergic receptors. You've worked on orphan receptors. Which one is your favorite?
I feel obliged to say the orphan GPCR that I'm working on for my PhD. I think Nicola would have my head if I didn't say it. That GPR 146 was my favorite GPCR.
Mhm.
But genuinely, I think GPR 146 is a really cool GPCR. It's an orphan GPCR, but I think in general, orphan GPCRs are so interesting. Um, it's a little bit of a trap though, because
Yeah.
they're very difficult to work with, and there is high risk, high reward. Um, but unfortunately, the risk often outweighs
Yeah.
the reward in orphan GPCR land. But yeah, my PhD is focused on GPR 146. Um, it is a severely understudied GPCR. Um, when you search GPR 146 on PubMed, you get something in the realm of 30 hits, um,
Yeah.
which is insanely low for a GPCR. Um, it's just there's there's quite a few GPCRs that fit the same kind of description
Mhm.
that are completely understudied, but have really strong phenotypes, at least in a mouse model. And I think that studying these receptors and finding a way to target them to manipulate those phenotypes is so interesting.
I think so too. And we do have that in common as well, because I too worked on GPR 146, trying to figure out if C peptide was a ligand.
Hmm.
And all the data with I was able to collect was inconsistent.
Yeah.
Um,
Yeah,
absolutely
no,
inconsistent. I was happy to
I've
see your data, thinking, okay, it wasn't me.
yeah, no, I've put some blood, sweat, and tears into GPR 146 and C-peptide, let me tell you. Yeah, I have, unlike you, have had consistent results. They've just been negative. I've just entirely failed to find a response at
Mhm.
GPR 146 testing C-peptide, which I don't think is uncommon, given that you found the same thing.
Yeah.
Um, but yeah, I think it just reinforces the fact that orphan GPCR research needs to be really tightly controlled, really tightly regulated, so that we don't have one study that then affects
Yeah.
10, 20 years worth of research down the line. And I think it really is our responsibility to um to show that those effects aren't true and to publish those negative results to put the nail in the coffin and to prevent other researchers spending years. And it sounds trivial to test a peptide at a GPCR, but at a
It's not.
GPCR where you don't have a positive control, where you don't know what it signals through, you don't know what assay platform to use. So you're essentially having to screen across literally everything, every signaling pathway you have, in what could be the wrong cell line. It's not so trivial as one would expect, and a lot of work can go into it, and a lot of wasted resources could go into it. So I think it's really important to publish those negative results. It's just unfortunate that we end up on these trails of negative results.
Yeah. And I think you make a great point around publishing the negative results. Unfortunately, I don't think there, well, not yet, there is a space in journals to publish negative results. And we've been, when I was a student, we were joking with my colleagues that we should create the Journal of Negative Results, because I think it should be an important thing. And actually, for GPCRs in general, we're thinking about how can we accommodate and become the kind of a repository for negative results, and then have that accessible to the community, and people can come and comment and have conversations, because I think it's important. I mean, you've
Absolutely.
spent a lot of time doing well-controlled experiments on GPR-46, and which reminded me, I tried to do cyclic KMP. We've tried ERK phosphorylation. We've tried microscopy. Nothing seemed to work, and we even tried playing around with magnesium quantities or concentrations,
Oh, wow.
and that was such a difficult, complex setup. Different sources of peptide, different ways of doing it. And it's unfortunate that, had we known, or had that data been available, then you know what not to do.
Exactly. Those efforts, that
Yeah,
money, those resources could have been used
exactly.
elsewhere. Yeah,
Exactly. And for me, it was a side project, so it didn't affect my trajectory. But think of, it's your PhD
mm,
project.
yeah, it's my sole focus. So
And yeah,
it is devastating, but I think we're getting better with publishing negative results. Um, I'm not saying we're there yet, but I know that negative results definitely end up in lower impact factor
Yeah,
papers.
yeah,
Um, but I think there are more people publishing negative results, which I think is a really positive sign. So if we could find some sort of repository that was well respected,
yeah,
um, I guess the fact is negative data is never going to be sexy. It's never going to tell
yeah,
an incredible story that people get excited by.
yeah,
It's just going to be a reference for what not to do.
yeah,
Um, and if we could improve the way that we communicate that I think it would be really useful for the
yeah.
scientific community, especially GPCRs.
I think so too. And I just thought of the name of that repository. So maybe it could be called Dr. GPCR Encyclopedia of negative results or something like that. Well, it's important. You never know. And I can't even estimate the number of PhD students and postdocs who ended up working on a project for a year or two, only to have somebody walk by them and say, oh, like 5 people tried this in the past 5 years and it's not working. It never worked.
Yeah, I had that realization when I gave the Dr. GPCR talk, when you mentioned that you had tested C-peptide at GPR 146. I was like, damn,
Yeah. Had you
there must
known?
be there must be so many other people around the
Yeah.
world that have tested this. They just haven't published it, or
Yeah.
they've
Yeah.
tested it but they don't have enough data to form a full publication.
Exactly.
And they're not going to spend any more time creating that
Yeah.
data because it is a waste
Yeah.
of time.
Exactly.
Um, yeah,
I know it's yeah, we need to find a way to communicate these things better. So
mm,
maybe it's something that we can work on our end in Dr. GPCR.
would love to see it.
Yeah. Um, what's next for you? You're finishing up your PhD. Have you thought about what's next?
Yeah, so I often find myself in this situation, but I'm stuck in the realm of whether to pursue academia or to go into industry.
Mhm.
Um, Industry's growing in Australia, but it's nowhere near the industry that's in Europe, that's in the UK, that's in the US. Um, so at the moment, I'm going to chase postdocs.
Mhm.
I've started contacting people overseas, and I'm organizing some lab visits, which will be really exciting. Um, so hopefully I can stay somewhat related to orphan GPCRs because they're great. Um, but yeah, I'm hoping to do the traditional overseas postdoc for a couple of years and then return to Australia
Mhm.
and really forge forward in some sort of research capacity, whether it's academia or industry. There's a story yet to be told there.
Have you thought about doing an industry postdoc?
That is
Which
something that I have considered.
Mhm.
Um, I think I would prefer to chase academia first because I do have a bit of a penchant for teaching.
Okay.
I've done quite a lot of teaching here at UNSW. I teach into pharmacology, I teach into biochemistry, molecular biology, I teach into the medicine program.
Mhm.
And I really enjoy that side of it, and I think I get a lot out of that.
Yeah.
So a, a combined track uh, career for me, I think, would be ideal. I know there are so many difficulties surrounding that, and I
Yeah.
think if I move into industry, I'll lose that education focus, um, which I'm not sure that I'd like to part ways with yet.
Mhm.
So I'm sticking with academia for now, but industry is not ruled out, that's for sure.
I think it dep I think if since you know what you want, try and think about, is there a possibility to do, maybe not now, after your academic postdoc, to do a 1 year short postdoc in industry, just to give
Absolutely.
you that opportunity to figure to see how things go on the other side of the medal, because I
Yeah,
think it's one coin and it's just the other side of the coin. And I think sometimes having good relationships with professors at universities would allow you to be a lecturer and teach
Mm.
while doing potential in industry postdoc. And I think everything is negotiable. And everything is nothing is impossible. You just have to work for it. But first you need to figure out what is it that you want.
Yeah, I'm hoping that my postdoc through academia, through a university route, will in a country that has larger industry opportunities. I'm hoping that that will grant me a few more connections and I'll try and gauge where I'm at there. But yeah, it's complicated and I don't really expect that anyone knows exactly what they want to do
Yeah.
in their final year of PhD. But yeah,
Well, if you browse the ecosystem, then you can find a lot of people in there and connect with them.
yes, I have seen a lot of postdoc
Mhm.
opportunities through the jobs
Yeah.
page. So yeah,
Well,
it's a great platform.
thank you. Thank you. All right, last two questions. Top three aha moments that shaped your trajectory. I think we've talked about one at least.
Yeah. This was an interesting one for me to think about. I think the first one was changing my major and discovering pharmacology and physiology and the way that we can control physiology through pharmacology. I think me finding that was the defining moment in my career. Um, Else I would have ended up down some sort of chemistry route, which I don't like to think about where I might be now, but I'm very glad that I I I found the wonderful world of molecular pharmacology. Um, I think my second aha moment was probably the oral presentation that I gave at ASCEPT in 2016. And I think that's because I've often struggled with confidence and and I think most undergraduates would going into a national conference or an international conference where it's really nerve-wracking. You see all of these huge researchers and you're giving a presentation to a bunch of people who pretty much everyone in the room will have more knowledge on the general field than you would. So you are the smallest of the small and you're presenting your data. And that is just simply terrifying. But the the fact that I did that, the fact that I gave two really good answers to two Nobel laureates in the audience, uh, it was just it filled me with a feeling of pride and excitement and ambition that I didn't really previously have. So, and I think that's such an important thing for um, meetings like that to occur, that undergraduates, that honors students, master students, PhD students can present at so that they get that feeling and so
Yeah.
that they can interact with the scientific community to inspire them to keep doing what they love doing. And I still feel that every year when I go to ASCEPT, after a year of rigorously doing science, you kind of just burning the candle at both ends and
Yeah.
you get to ASCEPT and you see all the amazing science, all of the amazing people that are involved in the science, and it just reignites that flame,
Yeah.
or ignites a new candle to burn at both ends for the year. So um, yeah, yeah, it's been an interesting journey, that's for sure.
It's interesting, and I want to thank you for mentioning, the confidence aspect, because most most of the time it's our female guests who talk about lack of confidence and being afraid, and I think it's everybody has those moments independently of who they are and where they are, and it's just a matter of building on it and going at it anyway. And I totally understand the fear of presenting your research in front of two Nobel laureates. and a room full of experts, but then you did it and you came out of it, with your head held high and proud of yourself. And I think that's very important. And I'm sure that Bob and Brian also have some kind of an adrenaline rush before they go presenting. They just learned it by experience to control it.
Yeah.
The other thing I wanted to mention, I think that the GPCR field in general, people are kind, especially with students, with each other too. Not like it's not. I've heard stuff at conferences, but I think in general, especially when students are presenting or trainees are presenting, PIs are really proud and they like to engage and have productive conversations because it's also fostering the next generation of GPCR scientists, which is, I think,
Absolutely.
super important.
Yeah. I found that at our ASCEP national meetings that the fostering the next generation is so incredibly important. We have a student forum now,
Mhm.
as a special interest group, for all of our students to join and to in some ways have a mini annual scientific meeting
Yeah.
themselves. And we organize a symposium, we organize different events throughout the year. So it really is aimed at training us to be the next generation of researchers that will take over the committees and
Yeah.
everything to do with ASCEP. So I think that's really important.
I think so too. And I think the other point I wanted to mention, and I always had trouble with this when you go to conferences, typically people are separated by their rank, everybody below PI and then PI and above. And that's something that I never understood because I think we're all scientists. We're all some with more experience here to present our work. And that's something that we've been really, really focusing on at Dr. GPCR. Every time we organize an event, we don't, we allow anyone who wants to give a talk to come and give a talk or present a poster. The symposia throughout the entire year, you saw it last time. We had PhD students, we had postdocs, we had PIs, and I think the level was such a high level scientifically that you couldn't tell who was the PI versus who was the trainee. And I think that's important because then trainees get to understand and hopefully build confidence and say, I actually presented in the same session as XYZ, who has 30 years of experience. And we got into a great conversation. I think that's very important.
Yeah, and making those connections then sets you up to reach out to that person and
Yes.
to ask them if they have any positions available in their lab for when you finish your PhD. It just
Yeah.
opens up the network. I
Yeah.
think it's great.
Yeah. Last question. Any advice for young scientists aspiring to contribute to the field?
We've touched on this briefly, but I think as a young aspiring scientist myself, I would just say do it. I've wasted so much time angsting over a particular experiment or technique that I haven't done before that I just need to get into the lab and start, order the reagents, whatever it is. Um, this is something else that I have a lot of anxiety around, is that if I'm doing a new technique, if I'm performing a new technique, it'll take me a number of weeks to build up the courage to get into the lab and do it because I'm scared that it's not going to work or I'm not going to understand it. But over the past 2 or 3 years, I've become a lot better at that. And I've learned just to do it, just to get into the lab and do it. If it doesn't work the first time, then you learn from that experience and you can identify what went wrong, what the most likely thing to have gone wrong in that experiment would be. Um, and odds are it's probably not going to work the first time. And you change, you modify the protocol, you do things better, and the next time it's so much more likely to work. And again, it's a learning experience. To just jump in to just do the experiment do the assay. You learn so much from it, and don't waste time.
Yeah.
Spending weeks angsting over it. Just go and do it.
Agreed. I think Nike was right. Just do it.
Yeah. And the same goes for sending cold emails to
Yep.
professors or things like that, or asking for a plasmid or something like that, because odds are they're going to be supportive. They're going to share their resources to further the field, for example. Or odds are they're really excited to get to know you, to know your research, to have you be a part of their lab. I think it's such a great community.
Yeah. And I think just adding to the just do it, a quick brief story, and this happened maybe two months ago. So I typically send out email invitations for people to join me on the podcast. Not That doesn't mean that anyone who wants to join cannot come. It's just that we need to be proactive about it. And I really wanted to have Ray Stevens on the podcast. And I've been talking to people, I'm like, yeah, I'm not sure he's going to come and blah, blah, blah. So one morning I was like, I don't care. I'm just going to email him. And I emailed him. It was a very short and sweet email. 3 seconds later, I get a response and he says, hi, Yamina, I'm a huge fan. And I was like, oh my God, Ray Stevens is a huge fan of the podcast. And actually, Ray Stevens knows about the podcast and knows who I am. But then I was anxious about sending this email for months, not constantly, but every time I remember it, I was like, I really want to invite him. I wonder what he's going to say. And I sent it. And then 3 seconds later, I get a response. And actually, the man knows the podcast, which I think is a big deal. We haven't gotten
Excellent.
a chance
It
to
is.
record our episode yet, but it's just that moment when I saw the response, like, wow, that was really quick. And then I open it and it says, oh, I'm a huge fan. So you never know until you send that email, to get their plasmid, to get an introduction, to get a conversation going. You never know. Worst case, it's a no. But even worse than that is not sending the email and never knowing.
Exactly.
All right. Thank you, Brandon, for your time. I really appreciate it. This was
Thank you, Yamina.
such a fun conversation.
It's been a pleasure.
Thank you. I'm going to let you go to bed. Don't go anywhere yet. And I'll steal maybe 2 more minutes of your time. All right. Talk to you soon.
See ya.
Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest. We also want to say thank you to our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners as well: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join us today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, feel free to email us at any time at hello@drgpcr.com. Until next time, stay safe.